Hello students, welcome to Leela's tutorial. Today let us discuss about generation of transgenic mouse. What do you mean by transgenic mouse? It is a mouse in which a foreign gene of interest is introduced in order to study about the gene which is responsible for the human diseases or any other characters. Right? So here how you are going to generate this transgenic mouse? You have three different ways. Here in the first type, you are going to select a mice, the female mice, the healthy one, and from that you are going to obtain an oocyte, right? So the oocyte is allowed to develop, the fertilized oocyte is allowed to develop into 8-cell stage embryo. So now in the 8-cell stage embryo, you are allowing the retroviral transgene to get introduced, right? So it is insertion of the transgene using retroviral transgene. So here once the transgene is introduced into the 8-cell stage embryo, it is allowed to get developed. Right? So from the 8-cell stage, it is developed into blastocyst stage, like this stage. Right? It is allowed to develop into blastocyst stage. Then after that, it is implanted into pseudo-pregnant female mouse. So what do you mean by pseudo-pregnant female mouse? The female mouse is allowed to mate with the sterile male, and then the hormones or the drugs are given in such a way that their hormone levels are same as the pregnant mice, right? So here after implanting this blastocyst stage containing the transgene into the female pseudo-pregnant mouse, its body will accept that embryo, right? There will not be any rejection, right? Then after the gestation period, the pseudo-pregnant mice will give rise to a progeny or the offspring, which is nothing but the transgenic mouse, which consists of the foreign gene of interest, right? The transgene will be present in this, and this transgenic mouse will be screened for the conformation, right? They are screened for the transgene presence. So in the second type, again you are going to select the healthy mouse, right? The donor female is selected. From it, the embryo or the oocyte is aspirated. So when you are aspirating the oocyte, here you can see the fertilized oocyte. Into the fertilized oocyte, by means of micro injection, the gene, the desired gene or the foreign gene of interest is introduced into the male pronucleus, right? The foreign gene of interest is introduced into the male pronucleus of fertilized oocyte. So then it is allowed to develop, right, in the lab condition, that is in vitro conditions. This fertilized oocyte containing the transgene is allowed to develop into 2-cell stage, 4-cell stage, 8-cell stage, then into this blastocyst stage. So at this blastocyst stage, you can see a lining of blastomere cells and the stem cells containing the transgene or containing that desired gene of interest. So now this embryo, which is a developing embryo, it is implanted into a pseudo-pregnant female mouse, right? Then after gestation period, again you are going to get an transgenic mouse, right? It is allowed for the screening for confirmation. Then in the third method, again you are going to select the female donor mice, right? And from the donor mice, you are not going to take the fertilized oocyte at the early stage or 8-celled embryo. But what you are doing, you are not extracting or aspirating the embryo outside, right? You are maintaining the embryo inside the female donor mice only, but you are aspirating only the embryonic stem cells from that developing embryo, right? So these are the embryonic stem cells which have been aspirated from the female mice, right? So once you are aspirating the embryonic stem cells, you are allowing them to be maintained in the tissue culture media. Right? You are maintaining them and stabilizing them. After that, here you can see the target DNA consists of the desired gene of interest. Now this desired gene of interest is introduced into this embryonic stem cells. So here you can see I have marked a red color for the target gene which has been introduced into the embryonic stem cells. Right? So then now the embryonic stem cells consist of the target gene in them. For the incubation of these embryonic stem cells, right, and then stabilizing them, again you are introducing these transformed embryonic cells back into the blastomere stage. Right? You are introducing back into the blastomere stage of that mouse. You are injecting these embryonic stem cells into the blastomere. Right? So here either you can do it directly, otherwise isolate the blastomere or aspirate the blastomere stage, develop your embryonic stem cells and again introduce into the blastocyst stage, and then you are going to implant this into the another female. Right? So here if you are doing that, then the embryonic stem cells are cultured outside, then they are again introduced into the blastocyst stage or the developing embryo. So now you can see the embryonic stem cells consist of that target gene. Then this is allowed or implanted into the pseudo-pregnant female. Then this after gestation period, it will give rise to the chimeric offspring. Chimeric offspring is, it consists of two sets of genetically distinct cells. Right? So now this chimeric offspring is different from the transgenic animal. Okay, it is not the same. So here the chimeric offspring which has two sets of genes, here it is again mated with the normal wild type mouse. Once it is mated with the normal wild type mouse, then it is the breeding and selection to homozygosity of the mutation. You will be checking for the homozygosity of the mutation and then you will be shifting these animals for the research. Either it can be your gene knockout mouse or gene knock-in mouse, right, or the model animals or the mouse models for cancer. So like this they have various applications. So what is the advantage of generating the transgenic mouse? First thing is it is of very low cost. You can generate the transgenic mouse with very low cost and it is very fast process. So the short time is required. Within very short time you can produce many number of transgenic mouse because their lifespan is also very short, right? So their breeding is also very fast. That means within a short time you can get more number of offsprings of the progenies. Then next one is you can study about the gene. See, now you have introduced specific gene. So for this gene, what are the characteristics? What are the phenotypic characters or what are the genetically related diseases? Everything regarding this gene you can study by using these transgenic animal models. Then it is to understand the human diseases. Which gene is responsible for which disease? How it is affecting some other diseases or the syndromes. Then you have study of therapeutic agents, right? How the gene is responsible for these therapeutic agents which are used for the treatment or curing certain human diseases. So like this, these transgenic mouses have many more applications in animal biotechnology which are discussed in further classes. Thank you.